Speaker of the day, Nanna MacAuley. She's a professor at the Department of Neuroscience, and she's an expert in the brain and how fluids move around in our brain. And so I very much look forward to your talk, Nanna. Please go ahead. I will actually just change your pointer to a laser pointer. All right. Thank you very much.
Thank you, Morten. So thank you very much, Morten, and for the rest of the steering committee for inviting me to be here today. Not least because my research here on cerebrospinal fluid is a little bit on the fringe of the field of aging and drug discovery. And this doesn't actually mean that we don't want to be within the field. We are simply just not advanced enough in the field to get to the point where we can actually look at aging or look at drug discovery, because we simply don't know the basic molecular mechanisms, I'll make clear in a minute. So we would love to be able to get some drugs for these mechanisms, because that's going to help the clinic a lot. And we actually, there's very strong evidence, or not evidence, actually signs that the cerebrospinal fluid secretion is actually altered in aging, and that can actually affect our brain cognition. We just don't know how or whether it actually does it yet. So another few years, hopefully I'll be part of the community. All right, so I'm going to talk to you a little bit about brain water. And we actually have 80% of water in our brain. So for the general neuroscience field, I like to say that quantitatively, our research is the most important neuroscience. And this water is obviously not lying at the bottom of the brain as it does on this picture here. It is basically centered in the ventricles in the brain, these water-filled cavities within the brain, and also circling the brain, as we see here, basically protecting the brain from mechanical impact. And cerebrospinal fluid is also what is between the cells in the brain. So anything that has to diffuse between cells in the brain would have to go through the cerebrospinal fluid. So generally, we're all very happy. I assume you also are very happy about the water you have in your brain, because you would not be doing very well if you didn't have it. But occasionally, we have a little bit too much of it. And that's what happened during, for example, brain edema formation, which follows, for example, stroke, so either a blood clot in the brain or a hemorrhage in the brain. What can happen is an elevated amount of water, as you can see here, as a brain edema, where you see the ventricle is compressed here on one side. And the problem with our otherwise protective cranium is that the brain cannot swell beyond the confinements of the skull. So what happens when water enters into the brain under these circumstances is that the whole brain tissue gets compressed, blood vessels get compressed, we have less oxygen coming to these areas, and we get even worse conditions. Another condition you might be familiar with is hydrocephalus, where these water-filled ventricles get enlarged. And you've probably seen these terrible pictures of small children with these large heads, which will happen if it's left untreated. And what happens actually here, we actually have an age picture. This NPH is called normal pressure hydrocephalus. So that's what we see in aging patients that have enlarged ventricles, we see here. And even though it's called normal pressure hydrocephalus, there actually generally is a small elevation in the pressure. But we don't know the mechanisms of this. So generally what we would like to be able to do, like we can do in many other medical fields, is to be able to treat these patients with some kind of pharmacology. And we actually cannot at this point. So what is basically being done is either, in the case of hydrocephalus, there's put a drain into the ventricles that mechanically leads the excess water into the abdomen of the patient. And that works quite well initially, generally, most of the time. But the median lifetime of these drains are 2 to 4 years, after which the patient has to undergo semi-acute neurosurgery for the rest of their lives. They generally cannot avoid a drain like this once they've had it. So it can be quite impactful to the patient. For severe cases of brain edema, what the neurosurgeons are left to do is actually take a piece of the skull off to allow the brain to swell beyond its confines for some weeks, possibly even some months, until the brain tissue returns to normal volume and the bone flap is put on again. So these are quite rudimentary, very invasive, and also associated with a lot of different side effects. So ideally, we would like to be able to prevent water from entering into the brain under conditions of elevated intracranial pressure, where we have too much water in the brain. And the reason why we cannot do that at this point is that we simply don't know how this water comes in. We've known about cerebrospinal fluid for thousands of years. We've known about cerebrospinal fluid secretion, where it takes place, for 100 years. It's actually 101 years since the first publication, but we don't know how. And that's why my lab is really interested in trying to find out how this happens. So to go back to some very basic stuff you probably did on the first semesters of your medical students' studies, if you're a medical doctor, of course, is how water can get across the membranes. So water can cross the lipid membrane at a certain level if there is an osmotic gradient. And these particles you see here are supposed to be osmotic particles. So if there's more osmotic particles on one side of the membrane, water can go across osmotically. This can be increased in speed if there's aquaporins, these water channels left, which is just, it's like an ion channel, it just allows water to go through instead. But only if there is an osmotic gradient. And just to take you even one step further back, osmosis only takes place if there is an osmotic gradient across a semi-permeable membrane. And everybody, even physiologists and neuroscientists, not cysts and not, and like, yeah, we know that. But as soon as we come into the complex tissue, they seem to forget. So I'm going to show you this little beaker a couple of times during this talk. So some new players in how water can come across the membrane are these co-transporters. And now for a change, I'm in a field with some more cellular type of people, so you might actually be aware of these transporters. So these are coupled transporters. You probably know the sodium coupled glucose transporter or sodium coupled calcium transporters. So these transporters are coupled with a couple of ions, and they can transport ions across the membrane in the general direction of which the gradient of the ion of the transported molecules tells it to go. So some of these co-transporters can act like a mini aquaporin if you want. So if there is an osmotic gradient across the membrane, water can flow through this protein as well, but to a much smaller degree than an aquaporin. But what I especially want to talk about today is the ability of these co-transport proteins to transport water independently of the osmotic gradient. So how this works is basically that these transporters, they transport the different substrates, and again, in the direction of which the transported ions are directed to go. And what these transporters can do is that they can take a fixed amount of water with them in the transport direction. So when the substrates bind, water goes in with it and exits on the other side. So it's basically like the transporter taking a little suitcase of water with it every time it runs in that given direction. And this amount of water, this suitcase of water, is independent of the osmotic gradient. So we can have an osmotic gradient going in the opposite direction and water flowing down some osmotic pathways beside this protein, but it would still keep running and take a little suitcase of water with it every time. And this is what we call co-transporter water, because water is taken with the co-transporter. All right, so back to our water flux. So we produce roughly 500 milliliters, a full bottle like this, we secrete from our blood and into our brains every day. And the majority of this comes across this structure called the core plexus, which you may or may not be familiar with. So it's a small structure you see here at the base of the ventricles, these water-filled cavities in the brain. And they look a little bit like this stylistic picture you have here. So it's a monolayer of epithelial cells that are packed full of different transport proteins that by some concerted effort transport sodium and chloride and water from the blood and into the CSF, the cerebrospinal fluid, in the ventricles. And we're not going to go into detail with all these transporters, and there are many more than what I have drawn here. You might recognize the sodium-potassium ATPase, you probably all know. Sodium-potassium 2-chloride co-transporter, we're going to get back to that one. And there's an Aquaporin one here, and we'll leave the rest for now. So it's generally been assumed, if you read the textbooks on this topic, which are mentioned in many textbooks, but it's probably like this long, basically because we don't really know what happens. But it's assumed that it takes place like it does in our kidneys. So our kidneys, especially the collecting ducts we see here, look a little bit like the core plexus. It's also a monolayer of epithelial cells that water has to cross, now in the opposite direction, from the lumen to the blood, as we reabsorb the fluid from our kidneys, whereas in the core plexus, we go from the blood to the lumen. So in the kidney collecting duct, we have this wonderful setup where we do have the required osmotic gradients. We see an elevation in osmolarity from the lumen to the cell, and again from the cell and into the blood. We have aquaporins, these water channels, very conveniently located on both sides of the membrane to allow this water flux. We're talking 180 liters a day that have to be reabsorbed from your kidney back into the bloodstream. So it's a very high capacity water transport, and it works really nicely like this. So it was assumed, as I mentioned, that this is what takes place in the coral plexus. And the question then is, do we have the right transport proteins to allow this osmotic water transport? And do we have these osmotic driving forces to generate this flux? So when an Aquaporin 1 was found, we see here in the luminal membrane of the coral plexus, is found in one of the membranes, this orange one here. It was assumed, as it is anywhere where an Aquaporin is found in our body, that that would be this transport protein that would take care of all the water flux. It was knocked out, and it was found to reduce CSF secretion of these mice by 20%, part of which may be due to the fact that the blood pressure, the central venous blood pressure, actually fell by 80% in these mice. So blood pressure does have an indirect effect on CSF secretion, so it's actually highly likely that Aquaporin 1 is not particularly crucial for this CSF secretion. Also shown by a couple of human patients who do not have any Aquaporin 1s. They have a whole bunch of other things that doesn't work very well with them, but they have no neurological deficits. So no signs of any disbalance in the brain fluids. Another illustration that conventional osmotic water transport may not be what generates our CSF was discovered both in our lab and also 50 years ago when they were really good at doing these physiological experiments. So this is just in rats. So what we see here on the y-axis is CSF production rate, so how fast we produce the cerebrospinal fluid. And what we see on the x-axis is the osmotic difference. So on the right side of the axis, what you see here is where there's more osmotic particles in the ventricles than in the blood. So we have the right driving force to make water move from the blood to the brain. And what you see here is an elevation of the CSF secretion as a function of an elevated osmolarity in the brain. That probably doesn't come as a surprise to anybody. What was interesting, if we reverted this osmotic gradient so that we now had a higher osmolarity in the blood, so water should go from the brain to the blood under those conditions, we still saw a very robust CSF secretion from the blood to the brain, basically indicating that CSF secretion can take place opposite of the osmotic gradient. So here we see there's fewer osmotic particles here, and nevertheless, we have an almost undisturbed CSF secretion. So we were then interested in, where are we here on the x-axis? We could have several milliosmolar extra in the brain, like we do in the kidney, to drive this water flux, but we really didn't know. So we looked at the osmolarity of the CSF and blood in rats, in pigs, and also in humans. And what you see in red here is the osmolarity of the blood, and in blue is the osmolarity of the CSF. And as you can probably very clearly appreciate, there is absolutely no osmotic difference between these two fluids. So there's no osmotic difference across the coral plexus tissue, this monolayer of tissue that creates our CSF. So we are basically here around 0 milliosmolar difference, and yet we still have robust CSF secretion, indicating that we might have to look at these co-transporters, which are able to actually produce water independently of the osmotic driving force. So we set out to investigate whether these co-transporters could be involved in CSF secretion. So we started ex vivo, so we took the brain out of mice and took the coral plexus out of these mice. And you can probably barely see it from where you sit. It's circled in red here. So it's this little structure that's highly vascularized, so a lot of blood vessels going through, which is what you can probably barely see if you can see anything here. So this tissue we wanted to investigate water moving when we put an osmotic gradient on. So we basically soaked it in normal artificial cerebrospinal fluid, which is around the 300 milliosmolar that our plasma is. And then we added an extra 100 milliosmolar. So we're up to 400 milliosmolar. And for those of you that are clinicians, you would know this is pretty high. Your body would never see this. So when we do this, we see a drop here in the volume of the coral plexus. So this is when you go down in line, it's the volume gets smaller. So water leaves the tissue because it's a higher osmolarity on the outside. If we then do this with potassium chloride instead, so we take the same osmotic force, 50 millimolar potassium chloride gives us 100 milliosmolar. So the tissue sees the same osmotic driving force, but now we see an increase in volume. So even though water is being pulled out osmotically, the cells swell, the tissue swells. And to find out which transporter was able to do this, we started out, there's a lot of investigations going on. We ended up, I say, looking at bumetanide, which inhibits this transporter, the sodium, potassium, 2-chloride co-transporter. Transports 1 sodium, 1 potassium, and 2 chloride ions, along with water. So basically what we see here, when we inhibit this transporter, we now no longer see this increase in core plexus volume, because the transporter can no longer run around, so the tissue basically just responds osmotically and lose water, indicating that this Kcc1 is a transporter that's responsible. And this transporter is expressed in the lumen side of the core plexus, the green we see here. This is the side facing the ventricles. And this transporter belongs to the co-transporters that is able to co-transport water. We've established that earlier in the Xenopus oocyte system. So it could basically be a good candidate for this. So now what was interesting was, which direction does this transporter run? What am I actually going in the middle? I think I have, sorry, one more slide. I forgot to delete. Here we go. So basically, there's a lot of discussion of which transporters are expressed in this tissue. And finally, an audience that probably appreciates that it matters a lot which protein exactly is expressed and which isoform is expressed if you want to make any targeted therapy. So there's been a lot of discussion of this sodium potassium 2-chloride co-transporter, Kcc1, and it has some cousins that's called the potassium chloride co-transporters, and some people have found these in core plexus and proposed these to be involved in CSF secretion. So we were very interested in which ones were actually present to find out which ones would be involved. So we looked first at quantitative PCR of which ones of these transporters were here, and as you can probably appreciate, Kcc1 is robustly expressed, and one of these ones, Kcc1, is also present at the RNA level. So we can basically assume that these other three probably would not be expressed to a significant level. I often take this out of the talk, but I just want to emphasize that, again, we made a lot of effort to try to find out which transporters were actually expressed here, which ones could be interesting. And there's a lot of different antibodies for these transporters, but they're often not really tested whether they actually are specific or not. So we expressed all these different proteins in Xenopus oocytes and cross-checked for all the different antibodies, and you don't have to go through this in detail, just to show you the antibodies we used, we first made very sure they only could recognize one of the different isoforms. So with these antibodies, we could find Kcc1 and Kcc1, so basically the same as what we found on the RNA determination. So we have Kcc1 and Kcc. Those are the two potassium transporting proteins that are both actually able to transport water. To find out which side of the membrane they're on, Kcc1 stained nicely for immunohistochemistry, so we could place Kcc1, which you see in green here, we saw that picture before, on the luminal membrane. The Kcc1, all the antibodies we could get our hands on did not work for immunohistochemistry, so we had to develop a new method to try to find out which side of the membrane this protein was on, and thereby whether it could contribute to CSF secretion or not. So we basically injected biotin, which I assume many of you are familiar with. Biotin can bind to surface proteins if you put it on the outside. So when the core plexus was intact in the animal, biotin could only bind to the luminal surface of the membrane. So we then pulled out the core plexus, lysed it, and then looked at the ratio between what was in the luminal membrane versus what was in the entire core plexus. And what we see for Kcc1 here in green, it's the one that we see on the outside here, we see a number higher than 1 when we look at the luminal fraction over the total. E-cadherin you see in red here, that is a basolateral marker, which we also see up here. And when the number is below 1, that's when we have more on the abluminal membrane than the luminal membrane. And you can probably appreciate Kcc1 co-localizing very nicely with E-cadherin. So Kcc1 would have to be over on the basolateral membrane, where it cannot participate in secretion of water across the luminal membrane. And we were then now left with which direction Kcc1 runs. And you may not consider that something that one would play with for that long, but we actually just wrote a crosstalk that was published this week in Journal of Physiology, where all we discussed with this person that wanted to challenge us on it was which direction this one goes in. So a whole crosstalk just on this. So we're all very keen in my field to try to find out. Anyway, so Kcc1, sodium potassium 2-chloride co-transporter, is expressed in many of our tissues. And it always runs inwards in all the tissues I'm aware of. But the sodium and chloride concentrations are very high in the core plexus compared to other epithelia, and it's basically these gradients that decide which way the transporter goes. So when we looked into the ion concentrations of the core plexus epithelium and the cerebrospinal fluid, calculations actually indicated that it would go outside or outwards. So we investigated this in two different manners. One was we took core plexus out again from the mice. We loaded it with a sodium-sensitive dye, and we can basically here look at sodium exiting the tissue via Kcc1. So if we inhibited Kcc1 with bumetanide, what we saw was an elevation in sodium, indicating that sodium would go from the inside of the cell to the outside, and then when it was inhibited the transporter, it no longer could exit the cell. So therefore we could propose that Kcc1 ran outwards. Another way of establishing this is looking at the potassium ion with the rubidium as a radioactive congener. So we could basically load the core plexus with reactive rubidium, and we can see the rubidium then going out through Kcc1, and then measure how fast rubidium exits as a model of how Kcc1 works. And when we inhibit this potassium efflux with bumetanide, we see that it's almost gone. So potassium going from the inside to the outside is predominantly via Kcc1, again supporting that Kcc1 goes from the inside to the outside. And the last and most important thing we then did was, now we know that Kcc1 is expressed, we know it transports water, we know it transports in the right direction, but the question is quantitatively, how much does it contribute to CSF secretion? And we then established an in vivo model of determination of CSF secretion, which was certainly not easy. People were very good at it back in the 1960s and 80s, but just basically stalled because they didn't know the molecular players, which we do. Now we have more access to molecular biology and more knowledge of this. So we basically had to pick it up again. But there's nobody in the world that could really help us because nobody did this anymore. And we also learned the very hard way, and all clinicians know this, but most of us, the rest of us do not know this, that if these animals during these experiments are not kept at proper body temperature, are not ventilated properly, are not monitored their heart rate and the different blood levels of gases, you simply don't get the right values when you look at physiological parameters. So CSF secretion drops to less than half if you don't treat the animal properly during the anesthetized period where we do this. But again, we learned it the hard way, so now we're really good at it. So the way this works is we have an anesthetized mouse, or rats we're more into now, but this is a mouse, and we basically infuse artificial cerebrospinal fluid that's heated and bubbled and everything you can possibly do into one of the lateral ventricles. And this artificial CSF has a fluorescent dye in it. So it basically goes into the ventricle, through the ventricular system, and we collect it again down here at the cisterna magna, and then we look at the dilution of this fluorescent probe. Because whatever it has been diluted with, that would have to be newly secreted CSF. So if we know the pump rate, we can calculate how fast CSF is being produced. And what you see here in the middle is basically what an experiment would look like. So this is as a function of time, and this is the fluorescence ratio. So basically what we pick up compared to what we put in as a function of time. So we see a stabilization up here, and using these numbers, we could get a CSF secretion rate in mice of 0.65 microliters per minute. And we then, with this scenario, wanted to be able to infuse different drugs and basically see whether we could reduce CSF secretion. But to be able to do that, we have to make sure to demonstrate that we could actually change solution and then keep the same CSF secretion rate if it was a control solution we changed to. So a time control experiment here shows you that once we've established these physiological parameters, we can measure stable CSF secretion for 2 hours. And then we are actually ready to put in our drugs. And I'm just showing you one today, which is this bumetanide that inhibits Kcc1, the sodium potassium 2-chloride cotransporter. And what you can see here is that infusion of this drug reduced CSF secretion to approximately 50% of control, indicating that when we look at the core plexus epithelium, we don't have the same scenario as we do in our kidney collecting ducts, where we have many, many millismolar, several hundred millismolar to drive the water flux that can go through aquaporins, which we have tons of in the collecting ducts, and they're tightly regulated to ensure systemic body fluid homeostasis. But in the core plexus, we need a cotransporter to aid in this water transport because we don't have an osmotic gradient and we don't have an osmotic water permeability due to the lack of aquaporins on the other side of the membrane. So basically, we need co-transporter water to transfer this fluid in the absence of an osmotic driving force. So basically, at this point, we're interested in what can modulate NKCC1, because NKCC1 is not a good druggable target, because it's expressed in the neurons, which are obviously also in the brain, and this transport protein helps deciding the inhibitory action of our receptors. So we don't want to touch NKCC1 directly. So we would like to know how it's regulated during sleep, dehydration, aging, for example, or how it's dysregulated in a range of different diseases where we actually have altered brain fluid accumulation, to be able to, down the road, find some druggable target that hopefully is specific to the core plexus epithelium, so we don't touch everything else in the body. And with that vision, of course, of being able one day to go into these different diseases where there is too high intracranial pressure due to too much fluid in the brain and reduce the amount of water, at least in some of these diseases. So with that, I'd like to thank the contributors to this project and my group, of course, and the funding agencies, and thanks to you for listening in.
Thank you very much, Nanna, for a fantastic talk. It's really interesting, this very fundamental process that is, I guess, completely necessary for life, of course. We have a number of questions on Slack. The most upvoted one is from David Garcia. What do you think about the theory for the cause of Alzheimer's disease as a malfunction of the CSF drainage in the brain?
Well, that's a very big question. So he's alluding to the lymphatics, where we look at the clearance from extracellular space, possibly. As you may or may not know, this is a highly controversial field. And it actually is distinct from what I work on, luckily, because, I mean, when we look at how water comes into the brain, the lymphatics is really how water circulates in the brain before it exits. So I think at this point, there's people getting very different results on this particular thing. So I would like to actually be allowed to not have an opinion until there's more data out there showing us whether it really is diffusion or convection in this particular case.
And of course, Mike Nedergaard from our university is one of the experts in this field. I'm asking a question here. Great talk, Nanna.
Thank you.
There are specific diseases associated with water transport defects.
Whether there is.
Yeah.
I mean, that's what we think. The problem is it's very hard to measure. So it's a very invasive way of measuring it, so it's hard to know in people. But there certainly are some diseases where there could be a hypersecretion, where there's too much coming in. So definitely if there's cancer, if the core plexus, we can have this hypersecretion. Right. And one disease we're very interested in is called idiopathic intracranial hypertension, where young obese females actually have elevated intracranial pressure to the level where it's very high morbidity for them. And we don't know how. We don't know why they have elevated intracranial pressure. So we're basically making an animal model at this point to try to see whether we can then tease it apart. Because we have to take core plexus out, and we have to do this in basic experiments. Right. But then there is all in brain edema that could be hypersecretion as well, by some mechanism we don't know. So that is one part of it, and the other part is whether or not there's an elevated secretion, we would like to be able to inhibit the secretion just to basically get to a point of lower pressure, even though the secretion was not elevated to begin with.
Very interesting. We have one question from Brian Booth. I guess this is also maybe a little bit sort of, I'll just ask it and then you can comment. Are there any lifestyle interventions that may affect these processes?
Any what, sorry?
Lifestyle interventions that may affect water transport in the brain. And is it, in a way, also, maybe the question is it good to have a high circulation, like production of water and resorption? Or is there any knowledge about that at all?
I mean, you definitely want those two to match. I think that's the most important, so you don't change intracranial pressure. I mean, if one adhere to this cleansing of the brain, then of course you'd like to have enough water to actually cleanse it. Yeah. And I think that what could be interesting with Alzheimer's and also old age, where there seem to possibly reduce CSF secretion and possibly a reduced fluctuation as well, which may be what actually influences some of the cognition problems. And for the lifestyle, again, these idiopathic intracranial hypertension patients, the only way to cure them at this point is for them to lose weight. So weight loss actually also will decrease the... If you're highly obese and have this, it's the only thing. Very interesting. We have a question from Yasmin Bage. Thanks for a great talk. Aquaporin 1 gene expression is affected by age in
mesenchymal stem cells, and dermal fibroblasts. Do we know anything about this in the brain?
I mean, the patients that don't have Aquaporin 1 in their body, the null versions, they don't have any neurological deficits.
Huh.
So Aquaporin 1 is expressed in core plexus. That's the only place in the brain it's expressed to any significant level. But it doesn't seem to be that important. And also, if there's no osmotic gradient, an Aquaporin is not really used for anything.
Right.
So I think, personally, I think Aquaporins are overrated in the brain. Not in the kidney, but in the brain. But that's a personal point of view.
I mean, that makes sense, I guess. Maybe one last question here from Ashish Raiput. Does CSF's circulation decrease in old age? Is there any information in this regard?
So that there are indications that it decreased, but again, it's very hard to measure in people, and nobody that I'm aware of has measured it in rodent models, which we would like to do again, because I think this aging, it could be very influential, I think, for cognitive decline.
Very cool. Great. Thank you very much, Nanna.